This is a really great place. I've really enjoyed the past couple of days here. Just great students, and the postdocs are great too, and faculty are great. It's just a great place. So I really enjoyed this. I know this is Darwin week here, and I hate to put rain on your Darwin sunshine, but some of my talk might do that. Darwin was, of course, a great guy. But everything, a lot of stuff has happened in evolutionary biology since Darwin, which isn't always appreciated by most biologists, even many evolutionary biologists. Darwin taught us a lot about how natural selection might operate, but all the details have taken years and years to fill in. And I've always thought the most important thing about Darwin, I think of Darwin as the big integrator. He really was the first guy to integrate a variety of information from all over science to try to understand one general phenomenon, natural selection being one of those. Now, we probably all agree that natural selection is maybe the most powerful force in the biological world, but it is not all powerful. And there's this tendency for many of us in biology to look at biodiversity and just immediately go for an explanation based on natural selection. Some people spend years, their entire life, trying to explain various aspects of biodiversity in terms of adaptation. But what selection can and can't do is dictated by what I call the population genetic environment. And it has three dimensions. So mutation, of course, is the source of all variation upon which natural selection operates. Recombination reassorts variation within and among chromosomes. And random genetic drift is sort of the lens on the evolutionary process. This is not some mystical force that we've all sort of make up. It's due to finite numbers of individuals and populations, and it's due to the fact that genes are linked tightly together on the same chromosome and therefore not able to evolve independently of each other. Now, this is not a trivial issue because all three of these dimensions of the population genetic environment vary by about 3 orders of magnitude among species, and I'll talk quite a bit about the mutation rate. But the other thing we need to consider is that adaptations don't just come from nowhere. We also need to consider what I call the cellular environment, and it too has 3 dimensions. So one of these dimensions is the laws of biophysics. There's a lot of physicists moving into biology these days, and it's been often said that biologists have physics envy, but in the end, physics does win. We can't push an adaptation to the point that the laws of biophysics are violated. And one question is, how close do we ever get to that? Historical contingency, this is nothing more than Darwin's descent with modification. So selection operates with what you have. You have to have a certain starting point, and that dictates where you can go subsequently. And then molecular stochasticity. Just as there's finite numbers of individuals in populations, which causes noise in the evolutionary process, there's quite finite numbers of molecules inside cells. So many proteins in bacteria, for example, are only present in 1 to 10 copies inside the cell, and this varies stochastically from cell to cell. So this is a source of noise in the evolutionary process as well. So we need to consider all these things together when we think about how evolution can and can't proceed in different lineages. And I and a few other people are interested in developing this field that might be called evolutionary cell biology. So we have this sort of strange situation in biology, right? We have a pretty mature field of molecular evolution. One could say that about genome evolution. There's this emerging thing called evo-devo. But for some reason, we've jumped right over the cell, which is where all the action starts. So if we're going to move things forward in this area, we need to start with some very simple issues. We need to do some good comparative biology. So whatever your cellular feature you're interested in, how does the efficiency of its operation and its structure, the molecular level, vary across phylogenetic groups, and what is the source of that variation? And here we return to this issue of the drift of the biophysics barrier. Is natural selection, in fact, so good that it can move all molecular adaptations up to the point that there's no further room for improvement? Or is there something else that stalls the evolutionary process before we get close to the biophysics barrier? That something else is what I call the drift barrier, and it leads to what I call the drift barrier hypothesis, which will be the second half of my talk. This idea is very, very conceptually simple here. The idea is that once natural selection refines any kind of adaptation, whether it's at the molecular level or the emergent phenotypic level, once you get to the point where the next advantageous mutation has a selective advantage smaller than the power of random genetic drift, selection can't push you any further. You've reached the drift barrier. It'd be good to be further over to the right, but selection can't keep you there because you're just overwhelmed by the vagaries of random genetic drift, and you move back down. So this is one of the things we're going to explore in some detail. So I've spent a lot of time thinking about genome evolution, and this is clearly one area that a lot of biodiversity cannot be explained by invoking selection. In fact, the only way we can explain many things at the level of gene structural variation and genome architectural variation among species is by the inability of natural selection to modify things in a way that would be good. And so many of these things are very well known by now. So at the one end of the extreme of the tree of life, we have the simple Priuses of the genome world. So these are bacterial genomes, nice, sleek, elegant genomes, very little intergenic spacer, no intronic DNA, usually only one transcription factor binding site, in some cases none because they have polycistronic nature. And then we have this stuff down here which might be called the epitome of unintelligent design. So these are big, clunky genes that we find in multicellular lineages, land plants and vertebrates in particular. Tiny islands of coding exonic DNA, on the order of 150 bases, in this gigantic sea of intronic DNA. The average intron in human genomes is something around 4,500 bases. And unfortunately, these things all get transcribed together, and we have to precisely splice out the introns to get a mature messenger RNA. And the point that's emerged from all this work is all these embellishments that we add to our genes are dangerous things. So all excess DNA is dangerous. If you want to add an intron to a gene, now you have to reserve about 30 bases for the proper signature for the spliceosome to know exactly where to splice you, to cut you apart and put you back together. Without that, you'll end up with frame-shifted transcripts and so on. Premature, so these are 5' UTRs that we have on the front ends of our transcripts as big multicellular organisms. These are dangerous things to have because if you pick up a mutation that makes a premature start codon, and that will happen in about 1 in 20 mutations, then you're going to end up almost always with some kind of screwed up gene. That's a problem. Even non-functional DNA is a problem to have. Now, obviously, if you have non-functional DNA somewhere out in the intergenic region, you can't suffer from a loss of function mutation, but what you can suffer from is a gain of function mutation. You could pick up a transcription factor binding site that could cause your adjacent gene to turn on in the wrong place. So all excess DNA is dangerous, and this explains a lot of the variation that we see in genomes across the tree of life, and you get this beautiful scaling regardless of what you're looking at in the tree of life. So there's no sudden dichotomous break in the scaling of various aspects of the genome with respect to genome size, say, between prokaryotes and eukaryotes. It's a complete continuity of scaling. In fact, this scales right into bacteria. So it's as though some general principles are explaining variation in genome architecture. And so now the question is, can we go beyond these general principles that seem to explain variation in genome architecture? Can these be extended to higher order aspects of biology, say protein structure, even the structural features of cells? And I would have thought, in fact did think when I was getting into this new area, that the closer you get to the outward phenotype, the less things can be evolving in effectively neutral fashion. But in fact, there's been many, many surprises, and a lot of what we see, even at the cellular level, can't be explained by invoking natural selection alone. And here's just some quick examples. I've done some very nice studies on the wiring, so to speak, of regulatory pathways in a couple of species of yeast. Saccharomyces, of course, is one of them, and then the other is Candida in many cases. And there's many aspects of cell biology. Everybody has to do it more or less the same way. So we all have to make ribosomes, right, to translate our transcripts. But if you look in two different species of yeast, the ribosome looks essentially the same. You have to make about 120 ribosomal proteins or something to make a ribosome. But if you look at the way the transcription factors used to turn on the ribosomal protein coding gene, two species of yeast will not necessarily even be the same transcription factors. So you get complete regulatory rewiring. It's as though you had a car 50 years ago and you studiously looked at the engine and then closed it up and drove around for 50 years and something went wrong with the car, and you looked under the hood 50 years later and it's not the same engine, and you never knew anything had changed. So something is going on in the underlying regulatory machinery of cells that allows this complete drift between phenotypes that are otherwise constant in different lineages. Transcription factor binding sites, this has turned out to be a big problem. We would like to think you could just compare intergenic regions between different species and find the conserved motifs. Those would be the transcription factor binding sites. This has turned out to be extremely difficult. And probably the reason is that most transcription factors like to have a match of about, they have a favorite flavor of maybe 12 nucleotides. It only takes about 7 bases in a region to make a match to the transcription factor that's almost like glue. So you don't need another 8th or 9th one. This leaves lots of degrees of freedom for the actual motif to wander around from species to species. Enzymes, there's been a lot of work done on enzyme reactions, of course, and it turns out we have a pretty good indicator of how fast an enzyme could work if it was simply diffusion limited, if it was the perfect enzyme otherwise, and it was only limited by the rate at which it encounters its substrate. Most enzymes are about 4 to 5 orders of magnitude worse than what you'd expect here. So they're far from the biophysics barrier. I'm going to talk quite a bit about replication fidelity and how the mutation rate evolves in different lineages. We'll see there's about a 1,000-fold range of variation. This seems to be the only trait, not today, we know how it evolves across the entire tree of life. And then I'm going to start talking about a problem that hasn't been solved, but I think it's one of the biggest mysteries in all of biology. And this final line here is, why do species make their proteins out of different numbers of subunits? And most proteins don't operate as monomers, single proteins, but they involve consortiums making dimers, trimers, tetramers, and so on. We don't know why this is, and I think it may be simply neutral across various lineages, and we'll see why in a second. So suppose you're a really, this slide is here to make the point. So suppose you're a really naive, newfound biologist, and maybe you've arrived from another planet, so you don't really know much about biology at all. But you would look at these organisms up here, and what would you see? You'd see enormous beauty and enormous diversity. And there's Darwin's famous quote down at the bottom, and you would proceed for years perhaps trying to understand what the selective forces were that generated this level of variation. And that's a common sort of procedure when we study many aspects of biodiversity, regardless of the level, cellular up to overt phenotypes. These, of course, are what are they? Not sunflowers, snowflakes. I'm one of these alien biologists, I guess. These are snowflakes, of course, and this is a simple consequence of physics, this diversity. So these are biology snowflakes. So proteins in cells come in many different sort of generic types. So a small fraction of proteins operate as monomers. A single polypeptide change folds up and does its thing. But as we'll see, more often than not, most proteins operate as dimers or higher order structures. So you can have dimers, trimers. Tetramers are often nothing more than dimers of dimers, and the list goes on and on and on. And one question is, does this level of variation that we see across the tree of life, and we'll see it's quite remarkable, is this driven by a selection? So this, the same enzyme might be a dimer in one species, a monomer in another, and a tetramer. Is that driven by selection as though it matters? Or perhaps all these are just substitutable for each other and they carry out their mission, their metabolic mission, let's say, in exactly the same way. And there's other things causing this kind of consortium to arise. So no matter what area of biology you're in, this question is relevant to. So every aspect of the cell consists of things that are higher order structures. So the flagellum is one famous case, the nuclear pore, the cytoskeleton, almost all the metabolic enzymes operate frequently as dimers or higher order structures. Same is true of transcription factors and so on. No matter what you think about, this will be true. And there's been a lot of thought given to, or a lot of speculation, I should say, given to why this happens. Why would it be good to have a dimer as opposed to a monomer? One idea is you add things together and you can increase the structural diversity of your protein. And I should say that when I talk about dimers today and trimers and higher order structures, the catalytic site is not being made at the interface. What's going on with the dimers? Each of the subunits retain their own catalytic site. So you're not becoming a dimer so you can do something that you couldn't do before. One could argue that it's easier to fold, and you'd be correct, it's easier to fold 2 small polypeptide chains than one gigantic one, and so on. And the list goes on and on and on about potential adaptive explanations for this kind of variation, but one should also consider the possibility that perhaps nothing more is going on when one makes a dimer, evolves a dimer out of a monomer, that you're covering up some structural deficiencies on the surface of your protein that now is masked by another protein, creating an interface. There are problems with making multimeric proteins. One of them is a simple encounter rate problem. If you want to make a dimer, you have to find a friend to dimerize with inside the cell. So you have to up the concentration of the protein inside the cell just to up the encounter rate. So that's a metabolic cost to the cell. There are problems with sticky proteins. If you want to be a dimer, you have to have an interface that likes to adhere to somebody else, and there are some chances you'll adhere to the wrong somebody else. Many of us will suffer from this kind of problem later in life. So here's an idea, just a very crude idea to think about how compensatory mutations could play a role here. Many of you will have seen pictures of idealized proteins that sort of look like this. Everything's held together inside there with backbone hydrogen bonds. That's the way the protein's held together. And this would be sort of the outer wrapping of the protein, the hydrophobic residues on the outside. I used to be a limnologist, so I get to go think about water molecules. Again, these are water molecules, and really happy water molecules want to be involved in 4 hydrogen bonds. So this is sort of a network of happy water molecules around a well-wrapped protein. So there's the backbone hydrogen, one of the many backbone hydrogen bonds. And maybe this protein picks up a mutation, so it's not folding around in an appropriate way, and a backbone hydrogen bond is now exposed. It allows a water molecule to intrude into the protein itself, and this is not a good thing. The water molecules aren't happy now, and the protein's not happy. Everybody's unhappy. But evolutionarily, what might happen is that you pick up a compensatory mutation in the protein itself, and so one thing, it just might refold to cover up that defect, or this might become part of an interface, and the interface eliminates the exposure of that backbone hydrogen bond to water. So this just gives you a general idea of how compensatory mutations might be involved in the evolution of an interface that involves no direct selection for the interface otherwise. This is a plot now of, this is a summary of the multimeric nature of proteins in 4 different major phylogenetic groups. So what I've done here is I've gone through the entire protein database and tabulated, say, in all eubacteria, there's about 4,000 proteins. We know their structures. How many of them have monomeric structures, dimeric, trimeric, tetrameric, and so on? And you can do that with eubacteria, the archaebacters, unicellular eukaryotes. Those are, of course, mostly yeast, inverts, vertebrates, and land plants. And this is a strikingly different pattern than we see for genome architecture. Remember, with gene structural diversity and genome architecture, things get very, very complicated as you move in this direction. But with protein structure, you don't see proteins developing more complex or less complex structures as you move across the tree of life from relatively simple unicellular organisms to more complex organisms. Regardless of what group you're in, about 2/3 of proteins are multimeric. So only about 1/3 of proteins operate as monomers. Of the multimers, about 2/3 are dimers, and you can see that they're more or less exponential decays. They're negative exponential distributions here. There's a little bit of seesawing here. Oddmers are a little less frequent than evenmers, and there's reasons for that I won't go into right now. But the main point is you don't see a lot of variation in the structural use of proteins across the tree of life. Now, you could argue that there's real problems with this kind of analysis because it's completely dependent on the investigators in all these different areas and the genes and the proteins they like and what gets deposited in the protein database. So I decided to take a somewhat different look at this and make sure we're comparing apples with apples and oranges with oranges. And to do this, what we're looking at here are a particular set of proteins that have conserved functions across the entire tree of life. So these are the glycolytic proteins and some of the TCA, or the citric acid cycle proteins. Again, 5 major groups here. And all I've done is color code things. So I'm simply, for example, for phosphoglucomutase, what this is saying is there are some bacteria whose PGM is a monomer, others it's a dimer, others it's a tetramer, and so on. And what you'll see is there's no gradient. As I said before, there's no gradient in more complex proteins going to the right or going to the left. This is telling us even within individual phylogenetic groups, the same protein with the same function but in different species can have different higher order architectural features, as many as 3 or 4. And there's probably many, many more in these various phylogenetic groups. There's really limited amount of information we have on protein architecture. So the question is, what's going on here? Why do these patterns exist? And it gets even more interesting. Because if you were to take two, say, organisms with one particular protein that make a tetramer, in this case, a tetramer is a dimer of dimers again. This is a protein involved in lipid or lysine biosynthesis. Here are crystal structures of these tetramers, one for E. coli on the left, one for tobacco on the right. They're both tetramers. They're both dimers of dimers. But you can see they're using completely different interfaces. So this you could call a back-to-back and perhaps that a face-to-face. It's hard to explain these kinds of differences unless there was some kind of intermediate state perhaps where perhaps you had a state like this. Then it became a dimeric enzyme and then re-evolved the tetrameric structure but by utilizing a different interface. And this is seen a lot. Here's another example. This is a superoxide dismutase molecule. This is one of the enzymes we use to deal with problems of free radicals that result from metabolism. There's one up here for a photobacterium, another one here below for a cow, and they're using completely different interfaces. There's a nice paper from Anno Panchenko's lab from a few years ago, and they went through laboriously for all the protein families that had known structure, higher order structures in, and they found that 2/3 of all protein families that contain multimers had variation in what they use for the binding interfaces. So this is a dramatic example of rather fluid changes in the higher order structure of proteins across the tree of life. So I've spent some time trying to figure out how you would think about this from a theoretical point of view. And here's a fairly simple way of thinking about it. So imagine this is sort of a hierarchical, not hierarchical, just an array of different structures. So down here would be a purely monomeric enzyme. Here'd be a situation where you have a very weak interface, and so a very weak dimer. Now you've added the residues in there for binding interface, so you're stronger there and then stronger there and so on. So you can think of this array of possibilities. And really, those of you who remember your biochemistry will remember that nobody's actually a pure monomer or a pure dimer and so on, but because there's association, dissociation events going on all the time. But you certainly can be at the, very close to one extreme or the other. And so the way this model might work is what we would want to ask is, well, what's the, given a certain set of evolutionary events, what's the probability, if we followed some lineage over a long period of evolutionary time, what would be the probability, and just took, say, a snapshot every million years, what would be the probability of being in this state or that state or that state or that state? So this would be like a steady state evolutionary distribution. To know that, you need to know the transition rates between these. And to know that, you need to know something about population genetics. Now, it's fairly straightforward to think about this, especially in the neutral case, which I'll show you in a second. But each one of these transitions is nothing more than the product of the mutation rate from this to this times the probability of that mutation going on to fixation. So we know roughly what these coefficients should be. And there's an interesting prediction of this theory. If, in fact, there's this quasi-steady-state distribution of protein types across the tree of life, if you could actually lie down these protein structures on a phylogenetic tree, if things are in equilibrium, there would have to be exactly the same number of transitions in this direction from monomer to dimer as from dimer to monomer. That's a prediction of the model. Otherwise, you wouldn't. That's a definition of an equilibrium situation. So that's a prediction that can be made. And as I said, well, I just want to make one other mathematical statement here without going through the mathematical details. It's fairly easy to solve what these probabilities are within a linear array model like that. So if you wanted to know the probability that you're going to be in this state at any one point in time, it's simply the product of the coefficients on the arrows pointing up to it and pointing down to it. So if we know what those coefficients are, we can solve for the steady state equilibrium. We can do that fairly simply in the neutral case. So I believe if we want to invoke natural selection ever, we need to have a null hypothesis that we can reject. So we need That's one reason, even if evolution isn't neutral, we need to know what the neutral expectation is. So here's the neutral expectation of how proteins would evolve. So in this case, we know under the neutral theory that the rate of transition from one state to another is simply equal to the mutation rate. Kimura told us that many, many years ago. So all the coefficients on the up arrows are simply equal to the mutational rate of gain of a new adhesive residue. Now going in the other direction, these arrows have different values because here there's 3 bonds to break, here there's 2, and here there's just 1. So you have this series going downhill, and you can solve these by using that trick that I just mentioned and come up with the equilibrium distribution. And there it is, and some of you might recognize this as the famous Poisson distribution. And what's interesting here is there's only one parameter here that's governing everything, and it's the ratio of the mutation pressure up to down, which should be sort of intuitive. And if there's high pressure in this direction to make more adhesive proteins, you're going to evolve in that direction. And if it's higher in that direction, then you'll evolve in that direction. But depending on that ratio, you'll predict a different Poisson distribution. So that's a prediction of the model in the case of neutrality. Now, what happens if selection is operating? Suppose selection is favoring you to go in this direction or in that direction. What happens? Well, the same general principle applies, but now we have to multiply the mutation rates in both directions by the probability of fixation in both directions, which is no longer the neutral probability and constant. And this actually turns out to be another simple mathematical trick. So if we say S is the selective advantage of moving from here or to here to here, just as before I said the key thing was the ratio of the mutation rates up to down, now we need the ratio of the probability of fixation up to down. And Kimura gave us the nice diffusion approximation to probability of fixation years ago. And it turns out the ratio of fixation of a beneficial mutation to the deleterious mutation of the same effect is nothing more than this simple quantity here. This is E, the base of natural logs, times 4 NES. NE is the effective population size. S is the selective advantage. 1 over 2 NE is the power of drift. So this quantity here is the ratio of the power of selection to the power of drift. And so when you do all the math now, you get exactly the same result that I just showed you, except the key parameter, there's one key parameter, but no longer is it just the ratio of mutation pressure up to down. It's the product of that times the ratio of selective efficiency up to down. So this should also be fairly intuitive. It's still Poisson, and the disheartening thing here is, well, it's nice that it all comes down to one parameter, but the problem is there's a lot of biology in this parameter. Mutations in there, drifts in there, and selections in there. So this means it's going to be pretty hard to work our way around to figuring out the mechanisms that would lead to these different kinds of protein architecture. But the main point is it predicts these distributions. So even with constant selection in every lineage, exactly the same kind of selection, same kind of mutation, and same kind of drift, we expect to see variation among lineages in the state of the protein. So I'll conclude this first part of the talk with just some simple conclusions. The first one I just made. You expect substantial phenotypic variation among lineages, even if they're exposed to identical selection, mutation, and drift pressures. So that's something to worry about, because what we commonly do, we go out and look at different species and see different phenotypes, and we decide immediately that this is due to the different selection pressures. We need to be careful here, because diversity will still arise because of the stochastic nature of all these processes. Another thing to contemplate here, which is not fun to think about either, is this. The most common phenotype to be seen is almost never the optimal phenotype. And this is because if it's good to be way over there, but mutation's pushing you over there, the most common phenotype is not going to be over there. It's going to be somewhere closer to what mutation is doing, depending on the relative power of these two forces. And then finally, I want to emphasize that if the power of drift is in excess of the power of selection here, drift just wins. And we're right back to the neutral model, and this is what we've long-term called what Tomoka Oda called effective neutrality. So I think we now have a model for ways to think about protein architectural diversity evolution, but we still have a lot of work to do, and it's going to require a lot of collaboration between good protein scientists who are willing to do this kind of work. Usually a protein scientist will get really excited about one protein, solve the problem in one species, and just move on to another protein. So unfortunately, we don't have good comparative work in this area. Now, the second part of my talk is going to get now at this drift barrier hypothesis and how it explains mutation rate evolution. So I want to explain what the drift barrier hypothesis is in a fairly cartoon fashion. So again, imagine you have an allelic series going from left, a really bad allele, to the right, a really good allele. And so selection's favoring you to go to the right, mutations causing these different categories to graduate to a lower class or a higher class. Usually it's easier to break things, so there's going to be mutation pressure in this direction. And so now imagine what selection is doing is it's operating on this allelic series, and we scale the biophysics barrier would be the perfect enzyme out here. And what's going on here, each of these alleles is improving fitness by a certain amount, but you see you're running out of space for improvement. And what selection does, of course, is it compares you today to the preceding state. And as we run out of improvement, the selective advantage of the next allele in the series has to go down. And in fact, it's going down at an exponential rate here. And so what will happen if you have all this going on, selection will gradually push you to the right here. But if for example, the population size is 10 to the 4th, if it's haploid, the power of random genetic drift is 1 over 10 to the 4th. And selection will stall when the selective advantage of the next best allele is 10 to the minus 4th, when these two forces are equal. And so here you would hit the drift barrier at a population size of 10,000. If you increase it by, I guess that's 100-fold here, you get to move down here, but then you'll stall there. So there's always a drift barrier, but as you increase the population size, selection is able to move you more towards molecular perfection, so to speak. So if you were to study a population that did this and got down to here over time and just took a snapshot every 10,000 million years or so, you would see a long-term steady state distribution with a mean somewhere around here. It'd be better to be way over here, and occasionally mutation will make an allele over there, but selection can't hold on to it, and you drift back to the left. And occasionally it'll make a really bad allele, but that's eliminated easily by natural selection. So you reach a steady state distribution, and if the population size increases by 10-fold, you move over to the right. So that's the basic idea. And now we want to think about what this implies with respect to one particular trait. And if this is a very important trait in evolution, this is the mutation rate itself. And so the way we'll think about this is like this. Imagine you have a clonal population, and it's reached a sort of a steady state mutation selection balance. So on average, I guess there's a little bit less than one deleterious mutation per generation. And now one cell in the population picks up a mutation that turns it into a mutator lineage. So it's got a higher mutation rate than other cells in the population. Almost all mutations are deleterious. And this is going to be a problem for this cell because it has an excess rate now of input of deleterious mutations relative to everybody else in the population. So that's an excess rate of input. Each mutation, regardless of who it's in, has a selective disadvantage of S. So you can calculate, this is just like a nutrient budget in a lake. You can calculate the excess number of mutations that will end up in this lineage, knowing this and knowing this. It's just the ratio of the input to the output. So this mutator strain will have this excess number of mutations. Now what we're trying to find out is how bad is the fitness situation for this mutator lineage. So we have to multiply the excess number of mutations by the effect of those mutations, which is S. This is one of those great things in populations genetics where a miracle occurs in the math. In this case, there's an S in the numerator and the denominator, and the S goes away. So what this is telling us is for an asexual population, the selective disadvantage of a mutator allele is simply equal to the rate of increase of deleterious mutations, completely independent of the effects of those mutations. Because the higher the effect, the lower they're being kept in terms of numbers in this lineage. That's likely to be a very, very small number because there's hundreds of proteins that influence replication fidelity. And if you just change one amino acid, it could have a change on the order of 10 to the minus 6 or something. It's a very small number. It gets even worse. And the point here is that means that selection on the mutation rate itself is a very, very weak force. Selection is trying to drive the mutation rate down, but it's a very weak force. It's even worse. It's quite worse for a sexual species. And the reason is, for a sexual species, you never get to build up this load. So you have a mutator allele. It's making deleterious mutations just like this. And the good news for sexual species is that all your deleterious mutations quickly move out to another member of the population, right? That's what you do is if you're reproducing sexually. In fact, for unlinked mutations, it only takes 2 generations for any mutation that you encourage the production of to end up in somebody else in the population. So for a sexual species, the penalty for a mutator allele is, again, that's the rate of increase of deleterious mutation production. We multiply by 2 because you're feeling those mutations for only 2 generations. And here we multiply by S, but the S does not go away. So this is a recombination mutation equilibrium, not a selection mutation equilibrium. That S is a very small number. We have many measurements of S for a variety of species. It's on the order of 0.01 to as low as 0.001. So what this is saying is that selection on the mutation rate in sexual populations is on the order of 100 to 1,000 times lower than it is in an asexual species. All right, so one more theory slide and then some data. So what this is saying about how the mutation rate might evolve in a population over time. Imagine we have a population size with an effective size of 10 to the 5th, an array of alleles again, but these alleles have a particular effect now. They're mutation rate classes. And you're moving, selection wants you to have is favoring anti-mutator alleles, but mutator alleles are always being produced. Here we start out with a population with a really high rate of deleterious mutation. And you can see it eventually wanders down in this gray area and it stays there forevermore. Here we start in the gray area and we stay there forever. Here we start with a really low mutation rate, and you can see the mutation rate gradually increases over time. And this kind of thing has gotten people excited in the past because people have argued that the mutation rate is optimized to reach some intermediate level that maximizes the long-term rate of evolution. There's no beneficial mutations in this model at all, so that is not what's going on. All that's going on is selection can't hold onto you because you're below the drift barrier. We increase the population size by a factor of 100, and you get essentially the same result. It takes longer to play out because the mutation rate evolves to a lower value. In fact, we predict with the drift barrier hypothesis that there'll be an inverse relationship between the mutation rate and the effective size. And now we're going to consider this with some data. We've been doing these kinds of experiments for many, many years. They're very conceptually simple experiments. They're called mutation accumulation experiments. And what we do, this is a paramecium, and what we do is we start out with a single individual or 2 if it's a sexually reproducing species, an inbred line or a perpetually clonal lineage, and then we make a bunch of replicates, typically 50 to 100 replicates. And then what we do with the population, each of these isolated lines, they start out all identical, and then every generation we put them through a bottleneck of a single individual, and that shuts down natural selection completely. The only mutations that selection can eliminate here are ones that cause lethality or complete sterility. So we catch every mutation you can imagine in these lines, every base substitution you can imagine. We get gene duplications, whole gene deletions. We've even had chromosome arm deletions in a few cases. So what we do at the end of the experiment now, we simply sequence all the genomes of these lines. They started all identical, and you just look for the unique changes in each line, divide by the number of generations, and you got your mutation rate, and you have the whole mutational spectrum. So that's the basic idea behind these experiments. It's a very powerful way to get numbers that are critical to evolutionary biology, but otherwise not easy to get. For those of you who don't believe we capture everything, this is result after about 300 generations of mutation accumulation in C. elegans, the elegant nematode. That's what you're supposed to look like if you're elegant in a nematode. And some of these lines have bizarre behaviors, and after a few hundred generations, bizarre morphology. So there's a big rotund one. Here's one with a kinked tail, a protruding vulva. This is not good if you're a nematode. But they're they're quite content to just get a new bed of E. coli every day at optimal temperatures, and they can last for years this way. We've done this kind of work with, we're trying to figure out how the mutation rate evolves across the tree of life. So in eukaryotes, we've got quite a menagerie going on. We've done the plant, the other plant here. That's a diatom. There's a slime mold. We've done several yeasts now. ichthyosporians. It's a lineage basal to all animals. It's even basal to quinoflagellates. Paramecium, I'll tell you about in a second. That's our favorite organism, Daphnia, and we've done fly, worm, rotifer, and so on. We've also embarked on a rather large project that some funding from the Army enabled us to do. We're trying to figure out in about 30 species of bacteria the complete mutational spectrum. And for these bacteria, what we're doing, we're covering the full range of GC content in genomes, because there's some bacteria as few as 25% GCs and others as much as 75% GCs. And it's been a long-standing mystery as to whether this is driven by selection or if it's driven by mutation. It's almost certainly driven by mutation pressures. Everybody has AT mutation pressure. This is also the full spectrum, the full length. There's not a whole lot of genomic variation in length in prokaryotes. It's about a total of about 100-fold, not as big as in eukaryotes. So, at the end of these experiments with bacteria, we produce these what we call wheel diagrams. So this is an experiment with Bacillus subtilis. It went for 5,000 generations. There's 50 concentric circles here, so 50 genomes were sequenced, and all those dots are the positions of the mutations found in the different lineages. You count them up, divide by the number of lines, the number of generations, and you get a mutation rate of about 3 times 10 to the minus 10 per site. This is why we have to make these lines go out for very many generations and sequence the whole lines. We're trying to estimate a very, very tiny number. And we're very good at this now. We have very, very low standard errors on this. There's a lot of variation in microbial mutation rates. The other extreme is illustrated here. The most extreme is illustrated here. This is a bacterium related to mycoplasmas. It's called Mesoplasma florum. The only thing known about this bacterium, it was isolated from a petal of a flower off a lemon tree. That's all I can tell you about its biology. But it's got a genome size of less than 1 million bases. So this is the smallest genome you can grow on a plate. Very, very low GC content you can see compared to this guy. We had this experiment going. We had to stop the experiment early because the colonies were getting smaller and smaller and smaller, so we expected their growth rate had to be going down. They had to be getting sicker. And so we sequenced them in only 2,000 generations and found that number of mutations. It has a mutation rate of 1 times 10 to the minus 8 per site per generation. So it's almost 100 times higher than it is for Bacillus. Now, to give you an idea how high that rate is, the human mutation rate per human generation is 10 to the minus 8 per generation. So this guy's going at warp speed with a generation ticking off every 20 minutes or 30 minutes. So it's a very, very high mutation rate. And so we're trying to understand what explains this variation in mutation rate across the tree of life. To put things in historical perspective, there was this very inspiring paper written by Jan Drake in 1991, not that long ago, but Jan in this paper in PNAS summarized all the data that we had for mutation rates. And this is all based on specialized reporter constructs. And this is a plot of the mutation rate per base versus the number of bases in the genome. So it's a log-log plot. The slope here is -1. That means the product of the number of bases in the genome and the mutation rate per base is a constant. And that constant's 0.001. And Jan really likes this number a lot. And so what Jan argued was that all microbes have a genome-wide mutation rate of 0.001. In other words, 1 in 1,000 baby microbes has a mutation. Now, if you look at these data here, there's only 3 cellular organisms here. So 3 of these guys are bacteriophage. These are viruses of bacteria. And if you eliminate the bacteriophage, you know, no matter how good of a statistician you are, this is not going to be a significant relationship. Now, we do have some significant organisms here. We have the bacterium, and we have the yeast, and we have another one. This is Neurospora crassa, the famous filamentous fungus. But what we want to do now is figure out how general is this result. Jan has a great line at the end of his abstract in this paper. He says, because this rate is uniform in such diverse organisms, it's likely to be determined by deep general forces. And he never told us what those deep forces were, but I would like to believe they're nothing more than conventional population genetic forces. So here's the pattern today. I just put this together a couple of weeks ago. These are all our mutation rates in unicellular species using this whole genome sequencing approach. It's a log-log plot, just like over here. The slope is actually not significantly from -1, which will make Jan very happy. But if you take a closer look at these data, there's some worrying aspects. So all the blue points are eubacteria. There's Mesoplasma florum, the one I just told you about. If you eliminate that one bacterial species, you can see there's no relationship to the mutation rate and genome size in bacteria. These are unicellular eukaryotes. In fact, that is marginally below significance. So the overall pattern is largely because there's one guy up there, one cloud of points there, and one cloud of points there. And we worry about these kind of things all the time with non-independence of data in evolutionary analyses. It gets a lot worse when you put unicellular or multicellular eukaryotes here. So here, Arabidopsis is in there. There's several invertebrates in there. There's human, chimp. We're pretty much the same as a chimpanzee mutationally. And there's mouse, pretty high rate. But you can see it's running in the wrong direction. So we need, if we're going to have a general explanation of how mutation rates evolve, we need to account for this kind of variation. Now, it's difficult to think of any causal relationship between genome size and mutation rate per base. So just adding more bases to your genome should not change the degree at which this base, is the replication fidelity of it. And this could be nothing more than another example of two variables being correlated with a third and making, leading you down the road with respect to causality. What we think is going on here is illustrated here, and this is related to the drift barrier. If you plot the data for all these unicellular species with respect to the effective population size, in the cases we know this, you get this beautiful negative relationship. Actually, you get two negative relationships. These slopes are not significantly different from -1. You can see they're parallel lines. There's one line for the bacterial species, and there's another line for unicellular eukaryotes. So the slopes here are completely consistent with the drift barrier hypothesis. Things with bigger effective population sizes evolve lower mutation rate. This is paramecium down here. It's got a mutation rate of 10 to the -11. Incredibly low mutation rate. It took us 3 or 4 years of mutation accumulation in paramecium lines to find like 20 mutations after sequencing a dozen genomes. We're not willing to usually take these experiments out for much longer than that. So what about these two lines? Why are there two lines here? Well, why aren't they all unified? Well, there's a good reason for this. Remember, selection is not really operating on the mutation rate per base. The theory I just presented to you, selection's operating on the total number of mutations produced per genome that influence fitness. These guys have much smaller genome sizes than these do. There's a much smaller target for mutations that affect fitness than down here. So typically 1,000 to maybe 4,000 or 5,000 protein coding genes up here. Unicellular eukaryotes have on the order of 10,000. Paramecium has 40,000 protein coding genes. So it's a very large mutational target. It's got twice the number of genes as the human genome. So when you correct these mutation rates per base by multiplying by what I call the effective genome size, the number of sites in the genome that count, in this case the protein coding DNA, everything is unified on one plot. Here is the slope of -1. So this is the total number of deleterious mutations arising per genome. It gets as high as almost 1 per generation for humans and other mammals. And you can see that this unifies not only the results from the two classes of unicellular species, but now everybody's running in the same direction. The multicellular species are on the same plot here. This looks like it has a slope a bit below 1, but in fact, I won't go through the details, but in fact, there's much more of the genome that is under selection in multicellular species. It's not just in the protein coding DNA down here. And what effectively that does is pivots this line up to roughly there. And it's almost perfectly in accordance with the prediction of the drift barrier hypothesis of the slope being -1. So this is, to my knowledge, this is the only trait that we have a general explanation for evolution across the entire tree of life. The prediction is quite simple. The mutation rate per genome is inversely proportional to the effective population size. If we bring over the genome size over here, the mutation rate per base is predicted to be inversely related to the effective population size and the effective number of nucleotides in the genome under selection. It's a very, very simple prediction consistent with population genetic theory, I think. Now, I'm going to close with one other aspect of some biology of errors inside cells that we have been starting to work on. I used to be an ecologist, and most ecologists spend all their time worrying about, and I think justifiably so, about things happening outside the organism, right? You worry about competitors, predators, pathogens, finding mates, and so on. But if you start studying cell biology, you quickly realize we're kind of our own worst enemy. I mean, in some sense, the inside of a cell is like a little ecosystem, and all kinds of things can go wrong inside the cell. So, for example, if you just want to make a good protein, all these things have to go right just to get down to a proper protein. So you can't have mistakes in your RNA polymerase, your spliceosome, the thing that cuts out the introns. They have to be constructed without errors. You can't make a mistake in your messenger RNA because that will make a mistake in the protein. To make proteins, we have to have good ribosomes. We use enzymes called aminoacyl synthetases. They recognize usually the right tRNA. tRNAs have to, of course, recognize the right codons and so on. There's many places errors can happen just en route into making a proper protein. We've started to focus on the transcription error rate. And so here we're asking, before I talked about the replication error rate, how often do you incorporate an erroneous base in the DNA? Here we're asking, how often do you incorporate an inappropriate base in a messenger RNA? And one can easily argue that the selective pressure for accuracy at the level of transcription is way lower than replication fidelity, and we will predict very high error rates. And the way to do this is illustrated here. I'm just repeating what I did for sexual species for the selection on the error rate. So a given change in the error rate at the level of replication, remember in a sexual species, we have to multiply that by the total number of nucleotides, the effective genome size, the total ones that count, where mistakes will influence your fitness. We multiply by 2 because in a sexual species, they're gone, often to somebody else within 2 generations. Then we multiply by the effect of the mutation. And we can translate this directly over to think about the transcription error rate. So given the same error rate, what is the price you pay for a transcription error? Well, it's not 2, because transcripts don't get transmitted across generations. Most transcripts actually have half-lives that are much less than the lifespan of the cell. I put a 1 here to be very, very generous. Clearly a 1 is smaller than a 2, but this 1 should probably be maybe 0.1 or something. We don't multiply by the effective genome size because not everything in the genome is transcribed. We have to multiply by the effective transcriptome size. So this is clearly a smaller number than this, and probably a lot smaller. S is going to be the same because we're asking, at the end, when all this is turned into a protein, what is the effect? So that will be the same here. We don't have to worry about that. But then there's going to be this dilution effect down here. So if you're a haploid genome and you have an error in your genome, every protein that you make is bad, and it's going to be felt. But if you're a transcript inside a cell, and maybe there's 1,000 transcripts inside your cell from that same gene at the same time, then there's going to be a dilution effect of about 1 in 1,000. So all these 3 numbers are the product is going to be quite a bit less than 1. So we predict that a very high transcription error rate. So how to measure this? We like to, I prefer not to just do theory, but to always link it up with the data. It's always going back and forth in our lab. So we spent a lot of time trying to figure out how to measure the transcription error rate. And this is not an easy problem. So the naive approach would be, and that's how we started, and we quickly learned our lesson very quickly. You would just go out to everybody's RNA-seq data sets, right? Everybody's sequencing RNA from everything. You would compare those sequences of the genome, and then all the differences would be transcription errors. Well, that would be great, but in fact, 99.99% of the mismatches are sequencing errors. They're not transcription errors. So we had to figure out a way to get around this problem. And the way we get around it is the same way we do science. In most cases, we replicate our experiments. So what we do is we isolate the RNA from our bugs, and we randomly fragment it like everybody does when they sequence genomes these days. And then we glue random barcodes on the end of these. So we have all possible random 8mers, so 4 to the 8th different barcodes we can glue onto these. So they all get glued on randomly. What we're looking, there's a transcript with an error in it, and that's what we're looking for. And you can see that what this does, it means that every mRNA molecule will almost always have not only a unique shear point, but it will also have a unique barcode. And then the idea is to interrogate each of these molecules multiple times. So the way we do it is we glue them down onto strep-evident beads, like we often do in sequencing reactions. And then we do a round of reverse transcription. We turn these into cDNAs, and we put the first round over here. We pull the beads down with a magnet, and then we repeat the reaction, put those over there in the second bin, pull them down again, repeat the reaction. So we do this. We interrogate each mRNA molecule 3 times, and then we throw everything into one giant cocktail and sequence it all in a giant Illumina reaction, map the fragments back to the genome. And what we're looking for are situations like this. We call these families. So there would be the 3 replicates. And we find tons of stuff like this, many, many singletons. They're all over the place. Those are the reverse transcriptase errors, sequencing errors, and so on. What we're looking for is this. So if there's a true transcription error, it should be present in every one of the replicates that we interrogate. So that's what we look for. And that's how we count true transcription errors, and everything else gets thrown out. And almost everything else are these singletons, as I said. And so here are some data that we've recently put together using this method. So Mesoplasma, the bug I mentioned before, there's our famous bacterium and famous yeast, E. coli and Saccharomyces cerevisiae, and there's C. elegans. Recall that typically replication error rates were on the order of 10 to the minus 9, as low as 10 to the minus 11. You can see these are very, very much higher numbers. What I've simply done over here to make it easy to think about, this is the ratio of the transcription error rate to the replication error rate in these species. And you can see it ranges from about 1,000 to 100,000 times higher. So the transcription error rate is really very, very high inside this diverse group of organisms. There's no reason to think that better RNA polymerases can't be made, but the point here is that the pain you feel in terms of fitness deficit is so low here that selection can't refine RNA polymerase to the level it can a DNA polymerase. And to put a little more meaning on this, if you apply these to the average length of a transcript, what this is telling us is that about 1 to 5% of all transcripts inside a cell have an error in them. So like every ribosome in a cell with 120 proteins, every ribosome has surely got an error in it. So there's a lot of this stuff going on. Eventually we would like to measure the translation error rate, but we have no idea how to do that. I have no idea how you would resequence the same protein without destroying the protein. Yeah. So if anybody's got any good ideas on that, we'd be delighted to know. And we expect there the error rate would even be higher. So I'm going to end my talk here with a quote that I like very much from Herman Muller, a former IU faculty member. This was published in the American Naturalist in 1922. I mean, this is quite an amazing quote. Must we geneticists become bacteriologists, physiological chemists? I'm not even sure what a physiological chemist is. And physicists simultaneously with being zoologists and botanists. And I'm sure all the botanists and zoologists are saying, please, no. But I think if we're going to understand, we really want to understand evolution in a real mechanistic sense, we have to start integrating biophysics and cell biology with basic population genetic theory. Had the good fortune to have a lot of good collaborators on this work. This is a chunk of my current lab. These are the people who have been involved in mutation work, and we've had collaborators from various other places in the world. We're really lucky the mutation work couldn't be done without funding, so we've been lucky to have funding from NIH, NSF, and the Army. And then I'm pretty fortunate to have a job. And I occasionally do theory here at the Kavli Institute of Theoretical Physics. This is on the UC, University of California, Santa Barbara campus. The ocean would be right about there. This is a really good place to just think and do theory. So thank you very much.